Hello everyone, welcome back to Vidhavasini Academy. In this lecture, we will learn about Mendel's experiment and his law. Mendel conducted hybridization experiment on garden pea plant. The scientific name for the garden pea is Pisum sativum. In 1865, he published results of his hybridization experiment in a journal named The Proceedings of Royal Society of Natural History. The title of his paper was "Experiment on Plant Hybridization". Mendel conducted hybridization experiment for 7 long years. His work was largely ignored by the scientists at that time. In 1990s, his work was rediscovered by the three scientists. Mendel is also known as the father of the genetics. So why Mendel selected garden pea plant? Why he selected pea plant, not any other plant? So features. These are the features why he selected pea plant. First is self-pollination. Second is bisexual flower. Third is short life cycle. Fourth is easily raised, maintained, and handled. So what is self-pollination? Self-pollination means, for example, we take a plant and flowering occur in this plant. This is another plant and flowering occur in this plant too, of the same species. And here it's a male reproductive organ and a female reproductive organ. That is stigma-stylate ovary. So if the pollen grain of the male reproductive part sheds its pollen on the stigma of the female reproductive organ, then it is a self-pollination. If the pollen grain will shed on the stigma of the another plant of the same species, then it will be a cross-pollination, right? Self-pollination means the pollen grain sheds its pollen grain on the stigma of the same flower, then it is a self-pollination. Self means self-pollination. And second is bisexual flowers. So what is bisexual flower? Bisexual flower means male and a female reproductive organ found on the same plant, right? Short life cycle. So what will be the advantage for the short life cycle? The life cycle of the pea plant was very short. So Mendel can study as many as generations in a short interval of time, right? He can study as many generations because the life cycle of pea plant is very short. The other features was easily raised, maintained and handled. Pea plant was easily raised. It can be easily grow. It can be easily maintained and handled. Pea plant has a well discrete defined characters. So these are the features why Mendel selected pea plant for his experiment. So here is a list of 7 pairs of contrasting trait. Mendel chose 7 pairs of contrasting trait for his experiment. The first is stem height. Stem height, tall is dominant and dwarf is recessive. So what is dominant and what is recessive? We will understand this in the next slide. So first character is stem height. The tallness is the dominant characters and the dwarfness or the small is a recessive character. Second is flower color. Violet is the dominant characters while white is a recessive characters. Flower position. Axil is a dominant characters while terminal is a recessive characters. So what do you mean by axial and what do you mean by terminal? If the flower grow on the top of the plant or at the terminal position, then it is a terminal position of flower. And if a flower grow grows here on the branches side, then it is a axial position, right? So axial is dominant while terminal is recessive. Pod shape. Pod shape, inflated. Inflated will be dominant. So it is a inflated pod, and constructed means kind of this. This is a contrasted, constructed, constructed. Okay, constricted, and this is inflated. Inflated is a dominant characters, while recessive is a constructed characters. If this port colors, the color of the port will be green, then it is a dominant characters. If the color of the port was yellow, then it is a recessive characters. Seed shape, round shape, or the wrinkled shape. If the seed shape is round, then it is a dominant character. If it is wrinkled, then it is a recessive character. Seed color, yellow will be dominant and green will be recessive. So these are the 7 pairs of contrasting characters Mendel chose for his experiment. Procedure. What procedure he chose? He observed one trait at a time. He did monohybrid cross, he did dihybrid cross. So before going to the next slides, first we need to understand some important terms. So first we will understand what is allele. Allele is an alternative form of a gene. So let's understand this with the help of an example. Let's take a homologous chromosome. What is a homologous chromosome? Right? And this gene E is responsible for example, hair color. This gene is responsible for the trait. This hair color is a trait. This hair color is a trait, and this gene is responsible for hair color. This gene is responsible. So this E is a allele, and the small e is also allele. So allele is an alternative form of a gene. The gene, right? Wild type. Wild type means prevalent allele found in a population. Wild type, the more number of allele, the more similar kind of allele found in a population are the wild type. What is mutant type? Which is present less than 1% in a population, then it is a mutant type. What is dominant one? Dominant will hide the expression of recessive allele. Dominant will mask the expression of recessive allele, and the recessive will show its effect only in homozygous condition, and in heterozygous conditions its expression is hid by the dominant allele. So what is dominant and what is recessive? For example, this is a heterozygous. This is a heterozygous condition because a capital T and small t is present, then it is a heterozygous condition in which the phenotype of the plant will be tall, but the genotype is capital T and small t. So the capital T, the dominant one, will hide the expression of this small t. The dominant will hide the expression of this recessive character. The recessive will show its character only in the presence of homozygous condition. If it is present in homozygous condition, then the recessive will show its effect. Otherwise, the dominant will mask or hide the expression of the recessive one. What is homozygous? Homozygous means two identical allele present. Homozygous means capital T or small t. Identical allele present, then it is a homozygous. and if non-identical, or we can say a capital T or small t is present, then it is a heterozygous condition. Hope all the important terms is clear to you. If you have any doubt, please comment us, or you can message us. Next slide, monohybrid cross. Mendel took a tall plant and a dwarf plant. He then did his selfing. On selfing, he found F1 generation. In the F1 generation, he found all the plants were tall and the none was dwarf. He took tall and dwarf, but in the F1 generation, he found all the plants were tall. After selfing the F1 generation, after selfing tall plant with a tall plant of the F1 generation, he found 3 tall plant and 1 dwarf plant. So what will be the genetic basis for this monohybrid cross? He took male plant, tall male plant and a dwarf female plant. This is a parental characters. Then the gamete will form. One allele will come from the parent or the male, or the another allele will come from the female. After F1 generation, all the plants were tall. Then all the plants were tall. After crossing the F1 generation's allele, he found this ratio, genotypic ratio and the phenotypic ratio. So how this Punnett square is formed? So it is a heterozygous conditions, and the gamete found will be of two types, a capital T and a small t. So this is female gametes, capital T small t, and this is male gametes, capital T and small t. Capital T, tall, capital T, small t, tall, capital T, tall, small t and small t, that is dwarf. So he found 3 tall plants and 1 dwarf plant. So the phenotypic ratio will be 3:1. But the genotypic ratio will be 1:2:1. Why it is different? Because we found one homozygous dominant and we found 2 heterozygous and 1 is homozygous recessive. So the genotypic ratio will be 1:2:1 and the phenotypic ratio will be 3:1. So what is phenotype? Phenotype is the physical appearance, how we see or how it will appear to us. It is a phenotypic, or its appearance. It depends upon its appearance. So phenotypic ratio will be how this plant will look, but the genotype will be different from its phenotype, right? So what will be the Mendel's observation? In the F1 generation, Mendel found that all the tree plants were tall and none was dwarfed. As you can see in the F1 generation, all the plants were tall and none was dwarfed. He found that F1 always resembled either one of its parents, and the trait of the other parents was not found in this generation. As you can see, he took tall plant and the dwarf plant, but the character will resemble only one plant, and the character of the second plant is not showing in the F1 generation. In the F2 generation, he found that some of the offspring were dwarfed. This was not seen in the F1 generation. Only one of the parental trait was expressed in F1 generation, while at the F2 stage, both the traits were expressed in the ratio of 3:1, as we can see that. right? Mixing of these contrasting characters, tall or dwarfed, did not show either in the F1 or the F2 generations. Mendel also found identical results in dihybrid cross. Traits that appear in the F1 generation are called the dominant traits, while the other trait is called the recessive trait. Right? So what will be the interference or inference or the result of the Mendel's experiment? Some factors pass down. What Mendel conclude from this experiment, he found that some factors pass down from the parents to offsprings through the gametes. Now these factors are known as genes, unit of inheritance. Dominant. Dominant will express itself and the recessive remains hidden. Allele can be similar in case of homozygous or dissimilar in case of heterozygous. The allele of the parent pair segregate and the only one of the allele get transmitted to a gamete, as we have seen this in our previous slide, right? During fertilization, the two alleles T from one parent and small t from the other parents are united to produce zygote. That the one T and one capital T and one small t alleles or the hybrid have capital T and small t, right? Okay. So let's understand dihybrid cross. So Mendel took two characters in dihybrid cross. In monohybrid cross, he took only one character, that is tallness of the plant, tall or dwarfness of the plant, or the stem height. So in this section, the parental will be yellow round seed and the second parent will be green wrinkled seed. Green will be the color of the seeds and the wrinkle will be the shape of the seeds. So after the gamete formations, two type of gametes will form. Capital Y and small y. Capital Y and capital R, small y and small r. After F1 generation, the gamete will fuse and the zygote will form. In the F1 generation, all the seed were yellow round seed. Yellow round seed. And after crossing the after crossing F1 generation, he found four types of gametes. So how these four types of gamete will form? This is one allele, this is second allele. So if we take one allele capital Y and half, that is half of capital Y and half of small y. This will with the another allele. The another allele will be capital R and the small r. Capital R and small r. So half of capital Y and half of capital R will form 1 by 4 capital Y and small r. Half of capital Y and half of capital small r will form 1 by 4 capital Y and small r. These are the gametes formed. Small y and the capital R will form 1 by 2 multiplied by 1 by 2, that will be 1 by 4 capital R small y. This gamete, and the last one will be 1 by 4 small y and small r. These were the four type of gamete that will form, right? If we cross and the zygote will form in the F2 generation, these will be the ratio for the genotype and the phenotype. So 9 will be round, yellow and round. 3 will be yellow and wrinkled. 3 will be green and round, and 1 will be green and wrinkled. So how this will come? Capital Y, when the female gamete meet or the cross with the male gamete, this capital Y and capital Y, capital R, capital R. This capital Y, this capital Y, this capital R and small r. This capital Y, the small r, the capital R and the small r. This capital Y, the small r, the capital R and the small r. This capital Y, this capital Y, the capital R and the small r. And this go on. The Punnett score, the Punnett square will form like this. We will write female gamete on this side and the male gamete on this side, and then we will form a Punnett square. The genotypic ratio and the phenotypic ratio will be this. Then the 9 will be like this. The 9 will be capital Y dash. It can be capital or small, and R. It can be capital or small. 9 will be like this, and 9 will be capital Y. It can be capital. The blank space can be capital Y or small y, and the other will be small. The 3 will be like this. The another one will be small y, and it can be capital R and it can be capital R. 3 will be like this, and the last one will be like this. So 9 will be round and yellow, 3 will be yellow and round, 3 will be green and round, and 1 will be green and wrinkled. So how this ratio will come? Because this genotypic ratio is too difficult to learn. How we will learn this? So let's understand this with the trick, or you can say a formula. We also knew that the phenotypic ratio for the monohybrid cross was this. If we multiply monohybrid cross, this is monohybrid cross ratio for the one, and this is monohybrid cross ratio for the another. So monohybrid cross, if we multiply this one, then we will get the ratio of the 3 3s are 9, 3 1s are 3, this is 3, and 1 1s are 1. 9 is to 3 is to 3 is to 1. We got this ratio, right? And if you want to know the phenotypic ratio of the trihybrid, then this ratio will be multiplied with 3 is to 1. Then you will get the ratio for the trihybrid cross. So let's calculate the genotypic ratio for the dihybrid cross. So as we already know, the ratio for the monohybrid cross was 1 is to 2 is to 1. So let's calculate the genotypic ratio for the dihybrid cross. So we will multiply 1 with this 1 with this 2, or if it is not, if some students will find it difficult to understand, then I will clearly show how this will happen. This is the genotypic ratio for the monohybrid cross, and this will be the genetic ratio for the monohybrid cross, and we will multiply like 1 multiply with the whole. This is the simple maths. This is the simple maths. 2 whole will multiply with 2, and this 1 will multiply with So 1 will multiply with 1, the ratio will be 1. 1 will multiply with 2, the ratio will be 2. 1 will multiply with the 1, the ratio will be 1. 2 will multiply with 1, the ratio will be 2. 2 will multiply by 2, the ratio will be 4. 2 multiplied by 1, that's 2. 1 multiplied with everyone, the ratio will be this. So you get the ratio 1 is to 2 is to 1, 2 is to 4 is to 2, and 1 is to 2 is to 1. So if you want to calculate the genotypic ratio for the trihybrid cross, you have to multiply all this with the 1 is to 2 is to 1. You will get the ratio for the trihybrid cross. Okay. So this was the dihybrid cross for the Mendel. Now understand Mendel's law. Mendel gave two laws. First was the law of segregation and second was the law of independent assortment. For a particular trait, each individual possesses two alleles. As we already saw in the monohybrid cross, for a particular trait, like this is the tallness, this is tallness. So for this particular trait, tallness is a trait, and this particular trait individual possess two alleles. At the time of formation of gamete, alleles separate from each other. So we already seen here, at the time of gamete formation, the alleles will separate, right? At the time of formation of gametes, alleles separate from each other so that each gamete carry only one allele. The alleles do not show any blending. Both the characters are shown in the F2 generations and not seen in the F1 generation. Right? Law of segregation states that there will be no blending of the characters. There will be no blending of characters at the time of formation of gametes. Alleles separate from each others and each gamete carry only one allele. This was the law of segregation. The second law of Mendel was law of independent assortment. This law states that two pair of traits that are combined in a hybrid segregate of one of the characters is independent of the other pair of character at the time of gamete formation. As you can see this in the dihybrid cross, this yellow color is independent of the shape of the seeds. The allele for the color of the seed is independent with the shape of the seed, right? And so this law states that when two pair of traits are combined in a hybrid, segregation of one pair of character is independent of the another, right? So law of independence states that one pair of character is independent of the another pair of character at the time of gamete formation. The segregation will be independent of each other. So this was the two law for the this was the two law given by the Mendel. So if you like this video, please like, share and subscribe. We will discuss another aspect of genetics in the next video. Thank you so much for watching. If you like,